Good afternoon, dear Tamara, dear speakers, ladies and gentlemen. It gives me great pleasure to welcome all of you in this Tamara's second home for the last 20 years and to open this symposium. We are honored that Tamara's close co-workers and fellow passengers during the science world of her have accepted our invitation to join us here in this symposium. Our great thanks go to Professor Miroslav Radman, our today's keynote speaker. We, literally speaking, cut him off his busy daily schedule, and he accepted to spend a few hours with us. And last but not least, I'm really happy to see so many young faces here in this auditorium waiting to accept Tamara's scientific legacy and continue her work. And now I will switch a little bit for a few minutes to Slovene, because I'm afraid that in English some of my feelings and thoughts may be lost. But for those who does not understand Slovene, the translation is included in the abstract book. Ne spomnim se, kdaj sem prvič osebno srečala našo direktorico, profesorico Tamaro Lah Turnšek. Vsekakor pa je bil Nacionalni inštitut za biologijo leta 2007, ko sem se na njem zaposlila drugič v svoji karieri, zelo drugačen od tistega, ki sem ga poznala leta 1991. In z gotovostjo lahko trdim, da za tem, kar danes predstavlja inštitut doma in v tujini, stoji prav naša slavljenka. Kot jo opisuje eden od današnjih slavnostnih govorcev, je kot orkan, in to ne v slabem pomenu besede. Prihrumi iz nič in ko odide, nič več ni tako, kot je bilo prej. Velikokrat je temu orkanu zelo težko slediti, saj nosi seboj vse, izjemne znanstvene in razvojne ideje, načrt, kako te ideje izpeljati in kako jih umestiti v slovenski in globalni svet. In ne pomembno, kako vse to narediti s karizmo dame, z veliko začetnico, v sproščenem vzdušju, katerega svojim posebnim stilom vedno znova pričara. Vse to in ne nujno v tem vrstnem redu. Ne, z njo ni nikoli dolg čas, saj poleg vrhunske znanosti, kateri je predana vse svoje življenje, nista tuja ne zgodovina, ne umetnost, ne ples do jutranjih ur. In seveda, z njo tudi ni vedno lahko, saj ni tuja odločenost, lahko rečemo tudi trma, da od svojih ciljev ne odstopa. In če si ponovno sposodim besede že omenjenega govorca, mi vsi temu orkanu le sledimo, medtem ko nas premetavajo njegovi vrtinci. Temu razburkanemu življenju Nacionalni inštitut za biologijo posveča današnji dan. Vrhunski znanstveni sopotniki in sodelavci slavljenke bodo na simpoziju predstavili svoj znanstveni pogled na življenje. Tega poleg vsega lepega zaznamujejo tudi težke bolezni, kot je rak, katerega slavljenka raziskuje vso svojo znanstveno kariero. In ne nazadnje tudi smrt. O vseh teh temah veliko razmišlja prvi med današnjimi slavnostnimi govorci, katerega misli uhajajo tudi onkraj naših bioloških meja. Morda se zdijo teme današnjega simpozija kar malo črne, vendar smo tukaj predvsem, da slavimo. Še na mnoga uspešna leta, draga Tamara. And now I'm passing the microphone to the chairman of this symposium. Two brilliant young minds, Professors Kristina Gruden and Gregor Serša. They were both already awarded with the most prestigious Slovenian scientific award, and Gregor has been recently accepted to the Slovenian Academy of Science and Art. So, Kristina and Gregor, the microphone is all yours.
Thank you very much, Marina. I think, however, that the day should be directed to our honorary guest today, to distinguished Director of National Institute of Biology, Professor Tamare Lah Turnšek. We will start now this with this scientific symposium. It's divided in two parts. In the first part, we will have honorary lecture by Professor Turnšek and a keynote lecture by Professor Radman. And another talk, and then after a short coffee break we will have another 6 lectures. Before starting with honorary lecture, let me just allow a brief introduction of Tamara's professional career and her achievements. She started and developed her scientific career at the Department of Biochemistry and Molecular Biology of the Jožef Stefan Institute in Ljubljana, where she spent many years. She was also spending some of her career outside of Slovenia, including brief visits as a visiting research assistant at University of Bonn in Germany, a visiting research associate at University of Newcastle upon Tyne, and then also she gained her postdoctoral experience at several places at Department of Pharmacology, School of Medicine, Wayne University, and also she was visiting professor and director of Metastasis Research Laboratory at Albert Einstein Medical Center. So she was really developing a lot of fruitful collaborations all over the world during the years. We know her as the director of the National Institute of Biology, a duty which she performs very well, obviously, for the last 20 years. She has a number of other commitments, and she is very vocal in Slovenian science. My own experience, if I may add this, is that she is really very important for our efforts to make science an important issue in Slovenian society. She's very known for her views on that, and I really appreciate her efforts for that matter. Her research interests comprise the role of proteases and their inhibitors in physiological and pathological conditions, particularly in cancers such as breast, lung, and brain tumors, and especially in glioma. She was focusing on that for some time. She collaborated, as mentioned, with many different groups, also companies, for example, with Krka company for the production of diagnostic immunoassays, and so on. So the list is really extensive and very long. Maybe I should mention cancer stem cells, which is also the topic of this lecture. So without any further delay, Tamara, please, we are looking forward to hear the lecture, Dual Nature of Stem Cells in Cancer. Please.
Thank you. So, first, I have to say that I'm really very honored and pleased and thankful, especially to my colleagues, Marina and colleagues, that organized this symposium, which was, for me, first a complete surprise, but then I gladly accepted it because I would really like to see all my colleagues, friends from here and from abroad, especially who took an effort to come here and attend and contribute to this symposium. I'm also very glad to see so many students and young people, so that I hope we will be able to transfer some of our ideas, knowledge, and especially perhaps the passion for science and dedication to science to them, and to continue where we will sometime stop. Not yet. So I would not waste my time anymore, and so I would like to explore. So we had some troubles with this transformation, but let's say I will talk about dual nature of stem cells in this lecture. And we will start with the first human stem cell on Earth, which we believe was the Adam and Eve product. And it's a source of good and evil, like the Bible teaches us. But already Hippocrates, who first coined the name cancer, carcinoma, for this disease, also wrote and described the vital force called nature or physis as the internal driving force of vital functions, which is the best remedy that should be reinforced by doctors. So reading this, I was thinking that maybe it's a hint on the existence of stem cells already at that time, and a suggestion these to be used to cure an organism and cell therapy. So we go along this line, and first I'd like to introduce to you the stem cells. I think all of you know that we have evolved from embryonal stem cells, and that in adults, tissues and organisms, two types of stem cells exist. One are mesenchymal stem cells and hematopoietic stem cells that all dwell in the bone marrow. Now I'll talk about the mesenchymal stem cells, which are part of the tumor microenvironment when I look at the cancer, but otherwise they are used very much in regenerative medicine, preferentially to the embryonic stem cells or induced pluripotent cells, and I will believe that you will hear more about that at the end of the symposium. These are used because they are not tumorigenic in vitro, neither in vivo in normal condition. But what about cancer? Can they be used as potential cell therapeutics for cancer diseases? Indeed, as you can see here, these are used alone or equipped, genetically modified with some genes or some enzymes, and act like Trojan horses, because they also show tropism towards wounds, towards inflammatory tissues, as well as towards cancer. But can they be used as cancer therapeutics? So this is, as we look at that slide, really a good Dr. Jekyll. And now, who is Dr. Jekyll? I'm sure that you all know this novel by Louis Stevenson, and what he wrote is that Dr. Henry Jekyll and his alternative personality, which was Mr. Edward Hyde, is one of the fictional characters who occasionally feels he is battling between the good and evil within himself, thus leading to the struggle between the dual personality of Dr. Jekyll and Mr. Hyde, and I think we all have something like that in our personalities as well, but we'll see how that ends. So first, the properties of the stem cells, for the people who are not very familiar with that, are that they are cells that can replicate itself, which we call symmetric division, or they can differentiate into different types of cells, what we call asymmetric division. But as it turned out recently, this can also be a process that is reversed, and in 2012, the Nobel Prize was given for medicine to Sir Gordon and Jan Manaka for the discovery that the mature cells can be reprogrammed to become pluripotent cells. So it's a reversed process possible. Is it possible also in vivo? We believe that it is kind of similar processes going on in vivo in our bodies after certain condition, and this is what we call cell plasticity or transdifferentiation and so on. So, but as each cell, also the stem cell can be damaged, so it can become cancer cell, or the well-differentiated damaged cells can become stem cells, cancer stem cells. So cancer stem cells really exist, as a number of publications are telling us over past several years. And we have to really change the paradigm of tumor evolution in this respect. So asymmetric division occurs, and we've changed the paradigm from understanding tumor as a stochastic accumulation of mutations that then give rise to the different types of tumors, a mass of different types of tumors, or there is a hierarchical order where you can have stem cells as a master cells that have the program for development of the whole tumor, and indeed only these cells, by this theory, can form tumors in another site or another organism. These other cells then develop by this asymmetric division into a different subtypes of tumor cells. These cells can also acquire, by genetic or epigenetic changes, the properties to move, and they form metastases. And as you know, metastases are the cause of 90% of cancer patients' deaths. So these migratory stem cells, you can see them here. First, these are stationary, sit quietly in their niches, but then they acquire this property, genetic epithelial to mesenchymal transition, we call that, and they start moving. And where do they move? They get out of their niches and move to another niche in another organ to form metastasis, if and only if they find the right niche. So not every tumor cell can dwell in every organ, but they select the organs, or better, the organs select them. So it's what we understand as seed and soil hypothesis, the theory that was coined by Stephen Paget already in 1885. So seed is a stem cell or tumor cell, and the niche is the soil. So now we understand this, and I will switch now to the tumor that was said before, glioblastoma or glioma. Glioblastoma is one of the most aggressive brain tumor, and it originates from less aggressive glioma, or becomes glioblastoma de novo, so it appears very suddenly. The tumors are very angiogenic. The tumors have a lot of necrotic areas, so very heterogeneous, and they do not metastasize, but they really have the characteristic invasion type. You can see here a single cell can infiltrate the normal brain parenchyma and then form tumor. And this is because we cannot control this. So the therapies like radiation or chemotherapy or surgery is very inefficient. And that's why the death is really, the recurrence is really very often, or nearly always occurs in an average time of 16 months. So what do we investigate here in our department? We investigate the Evil-Mr. Hyde cells. These are glioblastoma stem cells. And as I already told you, they sit in the niches, and how this happens. So any cell in the brain, like neural cell, astrocytes, or their progenitors, can get damaged. And so can the mesenchymal stem cells, normal mesenchymal stem cells that dwell in the tissue, become damaged and form the so-called tumor-initiating cell, and this cell starts to proliferate because of this damage and produce more and more tumor cells with additional mutations, and one of them is tumor stem cells, or tumor propagating cells, or cancer stem cells. There are different names from these types of cells. And this doesn't develop immediately. It develops when the tumors start growing, and as you can see here, different tumor cells are distributing in the organ, like tumor, we call this organ, but under certain conditions, like hypoxic conditions, are just right that cancer stem cells' properties can evolve from the tumor-initiating cell. So this is where it sits in the niche. And this was a theory by Rolf Bjerkvik, and so I think in our work, we accepted the theory and started working further on that. So what are the niches? The niches are, as we believe now, formed along at the capillaries, so these are endothelial cells, pericytes, and here are these tumor stem cells coming to the niche by being attracted to the niche via some cytokines. And this is protecting tumor cells from, let's say, damaging radiation or other therapy. And indeed, as you can see here, primary tumor can be removed, and there's no tumor, but there's a minimal residual disease. A few stem cells remain because they are very resistant and protected by niches, and after a while, and here average of 16 months, a new tumor will arise. So this is a problem. Now, we are looking here also at, in collaboration with Ron van Noorden's group, the stem cell niches, and this is how they look in real tissue. You can see here these capillaries. These are smooth methyl actin staining here, and around them are stem cells, here marked by this CD133, who should be, or is actually a marker for glioblastoma stem cells, CD133. And then recently, Barbara, when she was working in Ron's lab, produced also these nice pictures where you can see the niche again, CD133, another stem cell marker, nestin, which may not be quite specific for only stem cells, and then SDF1, all were like stained around this niche. In addition to that, we had a lot of catepsins staining here, catepsin B, catepsin X, catepsin K, and one of you who know that, but the people who don't know how these enzymes are important, I will show you a very simplified picture. This is also the activity of catepsin B, I forgot that, this catepsin B proteolytic enzyme is active, and these catepsins are actually proteolytic enzymes or peptidases that cleave peptide bonds in other proteins, and this is a family of cysteine catepsins. Here's 11 proteolytic enzymes, and this nice graphics I borrowed from Jan Kokos that he really showed the 11 catepsins. They all have the trivial name by letters, but in fact catepsin B is a specific structure, different from catepsin K, different from catepsin X, for example. And recently, just we started to collaborate also with Janko in relationship of these enzymes with the cancer. And what is their function, we honestly do not know, but they are there and they should do something. And this is another challenge for our investigations. So from this, there are publications coming, and this is Ron being a co-mentor for Urshka and Vashendria, who is also in the audience, I think, that we have published on the catepsin K. This is completely new finding, and also catepsin K in glioblastoma was a completely new finding that we reported recently in the literature. The other new finding that I didn't mention, and I cannot go into that, is CD9. This is a new marker that could replace CD133 marker for stem cells, and this was also a very good and very well-cited publication. So go back to our story. Dr. Jekyll then created a serum or potion in an attempt to mask this hidden evil within his personality, how to fight against this. However, in doing so, Jekyll transforms into Mr. Hyde, a hideous evil creature who was really mysterious and very violent, and by the time Hyde grows in power and becomes stronger than Dr. Jekyll. So is this a visionary message from Stevenson that we are losing battle against the evil? More a philosophical question, but with respect to cancer, I do not believe so. I believe that we are slowly gaining this fight against cancer. So now, really, what happens with these stem cells coming together, the good and the bad ones? They confront, of course. Where do they confront? They confront in the tumor mass. And as you can see here in this picture, tumor mass is not only different types of tumor cells, as was shown before, by different colors, but they also have, of course, cancer stem cell. Here you can see sitting around the arterial niches, but they also contain many of the infiltrating stromal cells. We call them stromal cells or stromal cells, normal macrophages, immune cells, fibroblasts, and mesenchymal stem cells infiltrating into the tumor, going into the tumor. And here is where the cell can confront in the niche different types of stem cells, so good and bad one. So now, how can Dr. Jekyll become Mr. Hyde? How can these good cells change into bad cells? It is possible that transition to malignancy of the mesenchymal stem cells in vivo can be contact independent, so there's simple a transdifferentiation of the cells, or can be dependent on the contact, that only after the contact these two cells will change. In indirect or direct contact, this is still a question, and we investigate all of this situation actually. And I just show you a few slides illustrating these ideas. So here is, as I already said, mesenchymal stem cells show tropism, travel to the tumor which they feel due to the many cytokines that are secreted, so they go there, they feel like they have to help somehow. And here is a mesenchymal stem cells, but originating from different types of cells, as I said, but when it comes to tumor, it can, as it was shown, to transdifferentiate into the pericytes. Pericytes are cells that are lining the endothelial cells of the blood vessels, and they can also possibly, and this is not quite proven, differentiate into glioblastoma stem cells, or cancer stem cells in general. On the other hand, these cells also secrete many cytokines and different factors that cause a paracrine effect on the cancer cells. They can either promote and inhibit the tumor growth, which has been shown in many publications, and this tells us that we are still not sure what are the positive or negative effects of mesenchymal stem cells on the tumor progression, which was observed, depending, of course, different experimental designs, different types of mesenchymal stem cells, and so on. So the studies can sometimes not be really compared. So to continue along this line, now a very recent idea was, and I really helped to publish that because I like this paper, that brain mesenchymal stem cells are the other stem cell of the brain, and they are very important because they, as I said before, can transdifferentiate into parasites, and parasites can transdifferentiate, it has been shown before, into neural stem cells or into cancer cells, into cancer stem cells, and these cancer cells become cancer stem cells under hypoxic conditions, as we already said, and here they do whatever they do. They promote tumor growth and also then again transdifferentiate into other types of cells. So this is a very interesting idea that mesenchymal stem cells can also directly transdifferentiate into tumor stem cells. And what we found in our work, and this was the work by Mona Oliveira, who is a PhD student from São Paulo University, working with us already nearly for 2 years, she found also the cell fusion between tumor cells and mesenchymal stem cells. U87 is one of the glioblastoma cells, and the other one is U373 that are a little bit different, but when we put them together with mesenchymal stem cells, we of course stain them so that we can see them. And what we have seen here, they merge, they fuse. Even more so, if you look at the more details, one cell can be engulfed by the other cells and degraded there, what we call cell cannibalism. And this is what Mona observed in, let's say, this lineage, U87, up to 30% of cells do this, whereas when we took U373 cells, only 3% of cells would fuse with the mesenchymal stem cells. So you can see that mesenchymal stem cells can talk to another cell, tumor cell, in a different manner. And this is what Barbara Breznik actually showed, which I'm showing here. Barbara did put these two colored cells together with the mesenchymal stem cells in a direct contact, direct co-culture in 3 dimensions. So this is a 3-dimensional spheroid growing into the collagen or any other matrix. And then also what she did studying this confrontation, she put this into the brain of the zebrafish. And so then we measured proteolytic enzymes and all sorts of things in this confronted cell cluster. And what we have seen that mesenchymal stem cells inhibited the invasion of U87 cells. So if these are U87 cells, there's inhibition of invasion. And if you look at that, here, these cells are invasive, but when they are in the contact with the mesenchymal stem cells, they're less invasive, so they a little bit of shrink. This is shown also here. You see this cells is invasive, but it's more invasive in the co-culture, and this is also shown here. And now fish. Zebrafish is a very good model which we started to work with recently, and again here we got exactly the same results. If this is the brain of the fish where this cluster co-culture of the mesenchymal and U87 cells were implanted, here you can see, that invasion is not really going on, actually even proliferation, so there's decrease in invasion, whereas if in U373 cells there's increase of invasion, and you can see, you can see here, that there is this invasive U373 cells that goes along the spinal cord even, not only in the brain. We have also measured proteolytic enzymes in this cluster, and we have seen that these cells decreased proteolysis, decreased invasion, and along them also proteolytic enzymes were downregulated, whereas here in U373 cells they were upregulated. So invasion correlates with the proteolytic enzymes expression, which we expected, in fact. Now, I would like to just sum up this thing, and you see here, this is a graphic summary, MSC's decreased invasion and proteolysis of U87 cells and increased it in U373 cells. So at the end, I would like to comment a little bit of these results. What does that all mean? So mesenchymal stem cells, we think, are potential Mr. Hyde in cancer due to possibility of direct transdifferentiation to cancer stem cells or secrete cytokines that change their genes, and then you can have the really bad or worse outcome of these interactions. They also form these fused hybrid cells, and these are novel monstrous Mr. Hyde-like cells, I think. And MSCs can be used for therapy of cancer, but at the moment only as a Trojan horses, as when they are genetically modified or equipped with the enzyme that would cause apoptosis, death of the tumor cells, when these cells would come together to the tumor cells and kill them like these terrorists do. So the proteases are also important here, and we still see if protease inhibitors may be also useful. So at the end, I would like to thank, especially thank all my collaborators in the lab, especially the young students, postdocs, and I think these are very, very valuable young people that we would like to keep here and really transfer our knowledge to. And certainly also to the neurosurgeon departments and all the neurosurgeons there. And in University of Ljubljana, I collaborate with Brigitta Lenarčič and Marko Novinec, Rado Kumil and Janko Kos, and I thank also my foreign collaborators today, specifically Cornelis van Norden and Vashendria Hira, and many others which I'm not going to list here because we are really, I think, running out of time if I don't finish right now. So thank you for your attention.
Thank you very much for this interesting lecture and an excellent overview of cancer stem cells, which is clearly an important topic in cancer research. We did not anticipate any questions or discussion. The speakers will be available for that during the coffee break, so please hold any questions until then. So we are proceeding now with the keynote lecture, which will be delivered to us by Professor Miroslav Radman. To start with, let me allow a brief introduction of Professor Radman. He was born in Split in Croatia and educated at universities of Zagreb, Brussels. He did his postdoc in Paris, and he was a fellow at Harvard University. At the moment, he's a professor of cell biology at the medical school of the René Descartes University in Paris, and also director of the INSERM unit Evolutionary and Medical Molecular Genetics. Professor Radman is a distinguished scientist. He's a member of French and Croatian national academies, Academia Europa, World Academy of Sciences and Arts, and he's also an EMBO member. It should be mentioned that he's founder of the Mediterranean Institute for Life Sciences in Split, which is an important regional hub for science in Croatia, and also one of his affiliations. He is very well known for his work on DNA repair, replication, mutagenesis, genetic recombination, adaptive evolution, and so on. Perhaps three most important discoveries related to his work are the discovery of the SOS response in bacteria, the discovery of the strand-directed mismatch repair system in Escherichia, which holds relevance also for humans, and the discovery that the mismatch repair system acts as the genetic barrier in evolution. So we are looking forward to the lecture. Professor Radman will deliver a lecture entitled Chemistry and Biology of Aging and Age-Related Diseases. Please.
Thank you, Gregor. Great. Dear Tamara, dear friends in the room and colleagues, you know that I have no sense for formal things, but it is really an honor that you chose me to come from Split or Paris to share and to celebrate this occasion. You can well celebrate it because I have more years of emeritus, and I can promise you is much more hectic and dynamic than the pre-retirement, especially if you are careless and organize an institute that is private and you have to look for money and so on. But it is truly, psychologically, it's great to start at any age a new career, to have it in head. This is great. And the bad thing is that, as the organizers know, that under the last moment you doesn't know where am I don't know where am I. And so this is the promise for the emeritus status. I can promise you only in this lecture what I can, and that is the improvisation. Everybody knows that I have to improvise things. Otherwise I fall asleep on my own lecture. And last night I came at about midnight, and I thought, well, let me see what Tamara was doing last few years and so on. And then I discovered something, and I changed completely until 2 o'clock my slides. And I hope that this double improvisation, one last night and then second now, will please you. I will refrain, given the title, aging, and so I will refrain from temptations to philosophize, to say anything philosophical. So, except the one, my preferred slogan about that is that biological aging is cultural vandalism. When we could do the best, if just we were not aging. The children are gone, the old parents are gone, everything, have a nice situation. Now you could be most productive. And comes the biology and said, Alzheimer's stuff, cancer. It kills the most efficient, the most competent learning and productive human machines, kills them because of Darwinism, because the life expectancy was selected for biological reproduction. And we could, without culture, we could well die at 30. This would be very welcome for biological evolution because there'd be many generations per time. So in terms of human evolution, biological evolution is fueled by babies. The more babies, the more they will be exposed to life conditions and selections. But cultural evolution is fueled by adults and old in terms of chronological age. And there is a conflict of interest. I promise you no philosophy. Conflict of interest between biological and cultural evolution, and I leave you with this to dwell yourself. It is a serious one, as we increase in competence and so on. You just heard Tamara, and the survival curve, this would go necessarily, the acquisition of knowledge and experience, and then comes the biological survival curve. And it's cruel. And since our evolution is, of course, mainly cultural, I mean, who will wait millions of years that our brain gets a little bigger or whatever, I leave you with this. So, I do have to, all right, so, I do have to show you this because I should be allowed a little bit of egotism also. That I see many young people here, and I'll show you very quickly a few fixtures in order to tempt some of you to come, to try, and to see whether to work on crazy projects in terms that at least nobody else seems to be doing them, and in a place that is incomparable, that you will be invited because I have the deficit in good people at this point. So this was the. So who will interrupt me after 40 minutes? Please do. Yeah. Be cruel. Okay. This is the Villa Dalmatia called, but it really is called Tito's Villa because he was meeting here with his non-aligned countries leaders. He was the third man And Yugoslavia used to be significant. Now we cultivate insignificance. But this is what we have. This used to be the caserne of for 200 soldiers who were bodyguards of Tito, Garde Républicaine, Republican Guard. And when Tito was gone and everything, so I convinced the government that they should, with a huge help of my dear friend and regretted friend, Maria Lačević, in tandem we are doing this. She was more credible and I was more punching. And we got this ruin, and I should show you. That's how the main atrium looked. In November 2003, then came this. This is one of extremely rare occasions when I put tie. But it was this was like president, and there was a lot. I didn't know what I was doing. I was totally naive and stupid. They were giving me this refurbished building and said, "You want it private, non-profit, independent of us? Okay, find the money." And so that was like giving you a Rolls Royce and said, "No, but you don't have gasoline." So that was simply to make a little story. But a year later, this is how it looked. This is Jean-Claude Maille for immunologists, and this is Errol Friedberg for those who work on DNA repair, and this is Maria Lačević, who passed a year ago. She was a fantastic fighter and a great lady. They are irreplaceable people. And here what we have. This is this atrium, which you saw as a ruin. This is the practically hotel room, that meaning that you have a place to stay when you come. Okay? And this is the large lecture room. These are laboratories, the kitchen, and everything. Here I play basketball and go diving here and so on. And this is how it looks. It's a little bit like a joyful monastery. Okay, monastery without the behavioral warriors. And these are the people. The problem is really, short of a couple of senior postdocs and one very brilliant one. I have difficulty finding there locally the really motivated young students. These are good ones, but they are artists. They are matter. These are good ones, but they are physicists. So, and there I go after this. This is just to let you know where we're from, somehow geographically and feeling wise. I streamed into Ljubljana last midnight. As after 40 years of adventure in studying mechanisms of repair of DNA damage, correction of errors, editing of DNA after replication, and while trying to do the recombination, we switched to asking a question. Let us ask the question of robustness of life. Which life forms are death resistant? Because that's what I would like to be. Resistant to death. And in 2003, one postdoc started working on it, and I won't be dwelling on this. This is simply to tell you that it wasn't a megalomaniac idea, let me find the secret of all diseases and aging, and yet this is how I will be finishing my talk. And that experience I very briefly passed through forced me to turn the head in another direction. And I can tell you, I will be telling you only maybe 2 or 3 concepts in the time I have. And apparently it's not easy. It's very simple, it's conceptual, but it requires now from you to turn the head. You may not want to do that. Okay. So the questions are, this is what I want to do. The success of half-century worldwide investment in treatment of chronic or age-related diseases, let's be honest, is dismal. Is dismal compared to vaccination and antibiotics for infectious diseases. And you can immediately see that maybe the whole approach taken is wrong. Believing that you have to understand each of the diseases and all molecular mechanisms, all kinases and post-translational modifications, which is absolutely important, in order to treat it. Now, Jenner, end of 18th century, couldn't even cause vaccination, immunization, because the word immunization, that's how much they knew. In terms of high technologies, okay, and yet Jenner and Pasteur, with common sense concept and courage, made by far the most spectacular success of interventional medicine into public health. No high tech, no knowledge of mechanism whatsoever. So I'm trying not to be original, try to approach now, to copy them, to approach the age-related diseases, and you will see that they are simple. Their cause is simple. So what does time to cells in the organism to be the most effective cause of morbidity and mortality? With fifth power of time, no treatment increases it with fifth power. Well, modestly, what's the chemistry and biological clock for aging and disease? Because the differences between shortest lived and longest lived animal is 10,000-fold. So something is tuning the lifetimes. And what determines the individual predispositions to different age-related diseases, i.e. facing age, what determines our destiny? When will the weak link first break, and what is the weak link? And I should be able to, again, this is in terms of introduction, what was said, the billions are going because my colleagues are lying. They are saying that things are very, very complicated in aging and disease, very complex, instead of saying we don't understand it. That would be honest. It's arrogant to say, I don't understand it, therefore it must be complex. And that's why I'm showing this. Right? Oh, I see, there will be a chunk missing. No. This is, to summarize, to cartoon, the way cardiovascular diseases and cancers and neurodegenerative and infectious diseases kill us with age. And the modest project is, okay, I can't change the destiny because I would need to change the SNPs and so on. But what we can do is certainly delay the destiny, because certainly for cancer, we have accelerating the destiny by inflammatory agents, TPA, the tumor promoters, and there are anti-promoters that without promoters extend the latency period. Okay, so we can play on that. And if this common, as different diseases as these that kill by excess cells and these that kill by deficit of cells increase the fifth power age, maybe that indicates that there's a common cause. If that were the case, then if we identify the cause we can act upon the cause, then it would be easier to mitigate all age-related diseases together than any specific one. And I leave now simplification to somebody else. This is the review over here by Brian Kennedy, the director, ex-director of the Bach Institute of Aging and great specialist in aging. And this is how he summarized what's common in studying aging in yeast, so in mammals and in human progerias. And he came with Ross and tissue homeostasis. Thank you, Brian. It's great. Okay, so because we were already on the Ross way, and tissue homeostasis I added last night because just for you, personnellement, you know. And again, this is pretty. Can I not, by brute force? can we, please, because there will be more of these occasions. That's even not brute force. That's being smart. Yes. Thank you. Great. Don't be offended, please. I find that my illustrious colleagues forget this. We are obsessed by DNA, me too, first, for 40 years, by DNA, thinking that all the secret is here. And this is not just to be against, it just doesn't make sense, okay? Because, let me start. A cell without proteins, okay, you phenolize the cell, it won't destroy DNA, is inanimate, it's nothing, it's dead. A cell without DNA, any DNA, including mitochondrial, is called erythrocyte. And it works for 2 to 4 months, meaning that life is proteins. The boss is not the DNA. This is the boss. It used to be the boss is these are the workers. Well, you know, here we have when the workers determine the destiny of the boss, of instructions, by repairing, correcting, reading, replicating, and so on, then this slave is not slave but the boss. And so if this in this circle here, this is a proteins that run all life functions, either directly or by synthesizing molecules that do things. And including, of course, maintaining in integrity of the instruction of the genome and copying it into blueprints to produce themselves. In studying robust organisms, we saw that by damaging only the genome, you can introduce 2,000 double-strand breaks into a bacterium, Deinococcus radiodurans, and if you leave it 3 to 4 hours, it will repair all of them. Because the proteome during that terrible treatment was completely protected. Who knows how to repair 10 double-strand breaks, knows how to repair 1,000, it's just 100 times more work. But what we learned that that the quality of the proteome is the one that determines the life and death. Because unless you have residual, after an insult, a residual activity of the proteins to initiate the recovery process in order for first genes to be transcribed and to make this DNA damage response, which I called SOS, a million years ago, when I was still in life, then you will see that this, unfortunately, because this is easy, I mean, this was easy to study. Every protein is a different story. It's not like DNA, the same structure, just the sequence is different. Be it what it is, what we learned is this, apropos Ross as a common, that the initial impact of Ross upon the proteome is reduced by scavengers. This is where Deinococcus radiodurans, the unkillable bacterium by radiation, million rats, it survives 100%, invested in these sacrificial molecules. They are small molecules which attract the Ross upon them, and they get damaged, and this way, by default, proteins don't get damaged. Then there is defense, which is catalase superoxide dismutase. And then, this will be perhaps new to all of you, is working on the intrinsic resistance for proteins to become inox. Because there's a clear way. This is much more efficient, actually, than decreasing ROS, is to become inox, and I'll tell you what it is. This being done, whatever remaining proteome damage is there, it will be dealt by blue stuff, by elimination, by proteasome, which is clearance, or what is not cleared goes into the aggregates, which is basically garbage, which has some signaling, whatever, but we won't deal with details. I think it will be only one result that I want to show you from this important paper that takes time to be seen, but this will remind you of Leslie Orgel's error catastrophe theory, where Leslie Orgel said error rate in protein biosynthesis is such that 20 to 30% of all proteins in the cell, while everything is fine, carry some erroneous amino acid. So there's a millionfold difference in per nucleotide or per amino acid synthesis, DNA and protein. There's a millionfold higher fidelity of DNA replication than of protein production. And then there it starts the cycle of bad proteins making bad DNA making worse proteins, worse DNA, and that's how it goes to death. This is to tell you that the phenotype of misfolded proteins, because here we deleted in E. coli the ribosome-associated chaperone. This is the nurse waiting for the baby protein to come out and fold it. Deleted either that or a late chaperone. And you know what happened? The carbonylation. This means oxidative carbonylation, means that CH becomes C double bond O, which is irreparable oxidative damage. That it increased, it almost doubled, which is a lot. You will see why. What it increased is mutation rate, spontaneous mutation rate increased by tenfold with these tricks. We have many more to tell the same story. But what is interesting now, if we add this vitamin E, water-soluble vitamin E, both protein carbonylation and spontaneous mutation rate drop down here. Even in the wild type, it will drop. What this told us, is that the misfolding of proteins expresses phenotypically through oxidative damage. That being misfolded, there's always hope that the next chaperone will come and refold you. But when oxidation comes, I'll show you the scheme in order not to fatigue your brain too early and to kill my lecture. This is the summary of about 10 papers. And this I call Krishko because Anita Krishko is the key author, the hyper-brilliant colleague of mine. This is to show you that if this is gamma rays or UV, and we look at the breakage of DNA, that with these survival curves, and I'll tell you what they are, that the breakage of DNA is equal in this and this and this. It's not that the one that is hyper-resistant didn't suffer DNA damage. It suffered equally as the one that is sensitive. What is different is protein damage. This is carbonylation with increasing radiation, or later you see with increasing age. And in every case, we found no exception. If this is Deinococcus radiodurans, this will be its survival curve. This is carbonylation. If this would be E. coli survival curve. And this is E. coli, just not to compare very different species, where radiation resistance was selected in the lab. Irradiate takes a radius, irradiate even higher, irradiate higher, and irradiate higher, and you get a, in the lab, evolved E. coli that is radiation resistant. In every case, it was accompanied by the acquisition of protection of the proteome. There was no exception. And so it is if you plot here human lifetime, say until 80 or 100, and then this would be the nematode, and this will be the human, and all of that is summarized, I think, further away. It is banal. It's saying that this is how proteins have to work in a way to get together to arrange themselves, like in the cockpit of the pilot. If you don't make the mistake, you can go on, and that's why there's no mess. There's not too much mess in life. But when this is carbonylated, these are mainly lysines and arginines. They are highly hydrophilic. Carbonylated are hydrophobic, and this is a havoc. What Deinococcus and rotifers succeeded in becoming extremely robust resistant is to keep their proteome in this state even at high irradiation doses. And that's the secret of life robustness. Protect your proteins from rusting. I think this is an argument, whoever is interested in this, just take the reference, an argument that protein carbonylation in isogenic cohort of nematode is predictive of the rest of the life, isogenic, because that's how they reproduce. And you can have almost a factor of 2 selecting for fast runners who have lowest carbonylation. So not only carbonylation is not the poor Sadman, he couldn't make it through in 80s because biologists love to be serious by being negative. It's not the consequence of aging. It is predictive of death. So better than that, you can literally do. This hypothesis also had to include reversibility of aging. If it's only on the protein level, if the phenotypes arise and are worse and worse and worse and worse with age or with radiation, because there's more and more and more fraction, the fraction of proteins that is carbonylated, it increases, then it should be reversible by protein turnover. And as you know probably by now all, this is a demonstration with 2 isogenic mice, one old, one young. After 10 days of sharing bloodstream, the old one rejuvenates, and all of the diseases that have been tested, osteoporosis and heart hypertrophy and so on, came back. But it's not, this is compensation. This is reversion of aging and disease by compensation, because when you separate them, and now you see, aha, is the rejuvenated old mouse now young mouse? And so the real age comes back. So it was not erased; it was compensated, but it is reversible. And this is to show you that we have now the improved the technology that we can see and quantify carbonylation of individual proteins. All right? And this is a human liver. You can see how unequal are proteins in face of oxidation. And this is the concept that I want to show you before switching to the tissue homeostasis part. Whatever we use, by the way, means of mutations in ribosomes, that is high fidelity, low fidelity, deleting chaperone, overexpressing chaperone. We did all the homework. The producing misfolded proteins increases protein oxidative damage at constant ROS. And this effect is much bigger than you could produce it by increasing the ROS. And I think I have it here from my paper with Nystrom long ago. This is conceptually important for you, for those who are not interested in ever disinterested in aging and surviving. And this is a part of 2D gel, and this is labeling the carbonylated protein. In the control, at this part of the gel, there was one. You use hydrogen peroxide at saturating concentration. These proteins arise at saturating. This is at constant ROS, like here, the same ROS, but streptomycin. And what streptomycin does is and the only thing that it does is by binding to ribosome, it increases a lot the error rate. So during streptomycin-fed bacteria produce a lot of erroneous proteins. Now what happens to their proteins is this. So this tells you evolution was great, once again. When oxidative metabolism came into game, sneaked into the primitive eukaryotic cells, there was now, over 1 billion years time for protein. It had to survive. Good news was a lot of ATP. The bad news was a lot of ROS. And proteins were naive at that time. And you can see this naivety by seeing how quickly you kill archaea and bacteria that are strict anaerobes. And they are killed by ROS. We know that. So this shows you what evolution did in terms of acquisition of oxidation resistance, of making proteins inox, and you change them a little bit by these mistakes, and you see what disaster it happens. So this is the concept of competitive antagonism. When misfolded, proteins have, this has two destinies. Either chaperones come first and fixes it native form, and now it is inox because evolution is built-in evolution of resistance. If oxidation comes, then poor chaperone cannot do anything anymore. The carbonylation locks in. You can't bring back this structure here. We showed that. And therefore, the phenotype of misfolding requires oxidation. It fixes, it locks in misfolded structure, and that's why we see. Now, this is academic. What is real is that while these are random errors here with ribosomes and chaperones, what we call polymorphism, we are all loaded with so-called silent mutations. Silent because we are born healthy. We're lucky here to be born healthy, and therefore these polymorphisms in proteins, we call them polymorphism or silent mutations because they don't do anything. Well, they are silent when we are young, but they start to be loud as we age, as exponentially increases the oxidation, and we'll see what I mean by that. Here is the example. These are 3 isomorphs of alpha-synuclein, little aggregates in motor neurons of the Parkinson's disease. And this is purified proteins. And we just ask experimentally in the test tube, please show me your oxidation sensitivity. Okay? I'll irradiate you to death until the plateau, and a single amino acid substitution. Please don't be tired. A single amino acid substitution that predisposes people to Parkinson at 60 shows this increased, this is a lot, carbonylation. The one that predisposes at Parkinson at the age of 30 already is the blue one. It's only one amino acid substitution. It's silent for everybody until 30. And there comes this cartoon that this is if this is not a unique exception, then this benign academic polymorphism that we thought serves just that we are not all the same, boring looking, but different, actually are conditional mutations that require time or oxidation to become phenotypic. And this would be the three morphs. This would be carbonylation with time, just as it was with radiation. And this would be the function dropping down. And so, in a way, we should be able to look this way at the entire proteome and tell to the newborn baby, help maybe the revelation of this diagnostic by, say, irradiating or paraquat treating the umbilical cord cells, and say, well, this lady, this little baby, when it will be old lady or old man, will first be sick of a renal disease, and second is, I don't know what, that we will be able to diagnose at the protein level the predisposition to age-related disease. And that's where we got the money for. Okay, so the difference between inborn syndrome, and maybe I should stop here actually, inborn syndrome and inborn predispositions, because both are inborn, is that syndrome is simple. It's a null mutation with immediate phenotype. The baby is either dead or sick at birth. In the same gene, a missense silent mutation that we call nicely polymorphism is a conditional time oxidation dependent mutation that will require 50 years to become phenotypic, and progressively so with oxidation. Is that at least simplifying thing? Should I stop or be okay? And I'll show to Tamara what I'll tell her when she visits institute. This is to explain why big animals, huge, with Peto's paradox, why big animals, let's say, a whale has 1,000,000 times more cells at risk than mouse, and lives 100 times longer cancer-free than mouse. This is Richard Peto's paradox. And this explains it all, but I don't have time to tell. Okay. All right. And this is about, somebody gets a sepsis, inflammation. Inflammation is like a developer of the black and white photography. The longer it lasts, the more it will become black. And we are loaded with latent diseases, cancers, of course, but all kinds of diseases that when old people get sepsis, they come back. It means that all diseases were not cured, corrected. They were, by cellular parabiosis, as I call it, they were silenced. They were silenced, and now the inflammation with the release of metalloproteases, it isolates the cells, and Patrizia Paterlini right now showed that by looking at small number of cells in the blood, that for every patient where there was a local inflammation, she saw in bloodstream the cells. So, plating the cells after making the primary culture, you see the real phenotype of the cells, especially from old mice. Most of them are dead. There are cancers that would never appear if you left them there. So this is, I call cellular parabiosis, a phenotypic suppression. And that explains the Peto's paradox very nicely because of this. The bigger the animal, the lower the metabolic rate, the lower the ROS. And I must finish with the evolution. Why such a variation? Because in order to reproduce a complicated, big biological thing like us and elephants, it takes time. You can't, yes you can, but there would be no people around. We wouldn't be talking here. You have to survive. You have to be, with longevity, just sufficient to reproduce. Well, that's much longer for elephant and whale than for mouse. And therefore, the evolution had to solve the problem of the latency of age-related disease in aging. And who wants to come to study that at Medius? Please come. Thank you very much for the occasion to bother you.
We will finish off this part of the symposium by the lecture given by Professor Dr. Gregor Serša. Professor Serša is head of the Department of Experimental Oncology at the Institute of Oncology in Ljubljana. His research interest is experimental and translational oncology in the field of electroporation-based therapies and radiation biology. He's a long-time collaborator of Tamara, and now we are going back to cancer topic. The lecture is entitled Our Journey on Winding Road on Cancer Research. So please, Professor Serša.
Good afternoon, and thank you, organizers, for inviting me here to speak in the honor of Tamara. I had a huge problem how to approach this lecture, and I was trying to remember the time when we met first, and as I believe it was the time when I started working at the Institute of Oncology, and at that time we were involved in tumors, tumor biology, biomarkers and so on, and she was already a key player in that field. So the second keyword that I would like to stress here also is that things repeat, the history repeats in time. And if I remember my beginnings at the Institute of Oncology, my boss, Professor Plesničar, has at that time been the pioneer of irradiating tumors and giving BCG around the tumors, increasing the inflammation. And I will come back during my talk to this topic again, and you will see that we are doing the same thing, just in a different way. If I may, I will go through some of the points that are, in my opinion, important. The first thing is that time is extremely important. It takes a lot of time before you can bring an idea through all the winding roads to the application in the clinics. So those that have tried that they know they often fail. And if you look at the bottom of this timeline, you see funding, 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 funding. And this seems to be the key word as well. So let's be honest, money makes the world go round. And then the other thing that I remember in the beginning of our journey through the cancer research is that Tamara and Janko have organized the first experimental and translational oncology conference. And then in the next conference, and that was at 2002, Janko just remembered me, that was the second conference that we've had in Bovec. And here you can see Tamara is already leading the way through the conference, so she has been there always with us. Since then, I see many faces. We have repeated this conference, and this is basically the only conference in our, let's say, area which deals with this topic, the cancer research and translational oncology. What do we do now? The topics that we still tackle are biomarkers, preclinical testing, immunology, and we are strongly involved in electroporation. So what we do together, we don't really do that much experimental work together. However, we do some conferences, as I said. We do teaching in postgraduate courses, where Tamara and I have started the doctoral studies courses on tumor biology and experimental oncology. And then we are fortunate also to have our journal, Radiology and Oncology. So Tamara is involved in that as well. So these are, let's say, general topics where we work together, and we have acquainted and known each other through the many years that we are involved in cancer research. What are the opportunities? The opportunities that we see and we are trying to evolve now is the collaboration on the project application. money, 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 money. And then scientific collaboration, which is growing now, I believe, in the recent years, and mutual use of infrastructure, where we share the facilities in pursuing the scientific questions that are being shared between both institutions. And to be brief, not too long, and come back to immunology as I started before, this is the thing. When I started in immunology, I swore myself, never ever I will work in immunology field. It's too complicated, especially in tumor biology. And lately I've been pushed back into this field again through the gene therapy and IL-12 and all these new immunomodulators that are being used in the clinics and helping the localized treatments. So for those that are not familiar with the idea is that you can treat the tumor locally very efficiently, but those local treatments do not have effect on distant tumors. So as how to make local treatment a systemic treatment, that was always the question, how to transform that, how to combine that. And lately the idea came that local treatments can exert some local immune response that can be boosted by systemic immunomodulators, blocking some immune blockades within the tumors and within the organism, and giving the organism the chance to combat also the distant tumor cells and metastases. So as you can see this scheme here on the slide, the point is local tumor irradiation, local therapy, whichever, that induces the immunologic cell death, can be then boosted by some other adjuvant treatments. And those local treatments can be either irradiation or even chemotherapy or other ablative techniques like electroporation. So now this is my field of interest that I'm familiar with. We use electric pulses as physical force to destabilize the membrane and enable molecules to enter into the cells. And those molecules can be either chemotherapeutics or plasmid DNA. So we can do gene therapy by gene transfer, as we call it, or electrogene therapy. So if you prepare plasmid, which will encode some kind of a protein that would have immunological effect on the organism, you can apply it locally, and it will exert its effect locally and might be shed also into system, so it will have also some distant effect. So we have been working with electrochemotherapy, which is a local ablative technique, for many, many years. It's now in the clinical use. It's been in the guidelines, and you can use it, and 160 centers throughout Europe use it. So if we can combine this local ablative technique with gene therapy, that would bring into the milieu of the tumor also some immune response or boost the immune response, then we would be able to get the abscopal effect, or let's say transform local into a systemic therapy. So we have electrochemotherapy that basically kills the cells through the drug which comes into the cells. But in the end, there is some help of the immune response as well. Because if you treat the tumors in immunodeficient mice, you cannot cure the mice. You can almost eradicate the tumors, but the tumors come back then, compared to the immunocompetent mice. So this immune response, the immune competent component of the electrochemotherapy can be used and boosted by some other adjuvant treatment. You have some immune response. You can see inflammation in the patients where electrochemotherapy was exerted on those legs, for example, here on the melanoma metastasis of the patient. So this can be used to be combined with peritumoral gene therapy of IL-12, which is immunostimulant. So then these two therapies would act together, help local response, and also have some abscopal effect. The whole idea of combining this, we are just working in one way. The others are doing it in a different way with radiation and standard treatments. The point is that if the curve goes down, that means you have had, and you shift the curve, then there is palliation. There it's not curative effect. It is you prolong the relapse of the disease and the patients eventually die anyway. So all these treatments are very efficient in increasing the quality of life for those patients and prolonging their life as well. However, what we need to do and we want to have is the longevity. So that means you have to cure those patients. And if you cure those patients, then you don't have to have any relapse of the disease. So basically you have to cure the tumors locally and eradicate the very last tumor cell within the organism. So this is our goal. We can get some complete responses, as we call, with gene therapy, which can be increased. We believe it can be increased by combining local electrochemotherapy with peritumoral gene therapy with IL-12. We constructed this very complicated scheme. It's basically a model how it should work. I will not go into the detail. You can read it in the paper. But basically what we want to have is to boost the immune response and transform and translocate it in the lymph nodes adjacent where the tumor cells reside as well. We have some preclinical data where you can see that by combining you can get certain degree of the complete responses of those tumors that would not have been treated by either of the therapies alone. And then Maya is working in the vet clinic in Ljubljana, doing this in the clinical level in the veterinary oncology and being one of the best models also for the clinical application and translation into the clinic, human clinics. And you can see that these tumors are going away nicely, and these dogs are extremely long-lived after the therapy. They do not die as would have been expected without any other treatments as well. So where do our roads cross? They cross in many ways. We need biomarkers. We are coming back to this keyword again because many patients respond and many patients do not respond, and nobody really knows why this happens. And we need to know more about this, and we need to find and maybe together work on it into what is behind the whole thing. And this is our group that works on electroporation, as you can see. Thank you for your attention.